to all the participants for our second Zoom session for the course regarding the essentials of bioinformatics, or you can say the basics of bioinformatics. So before starting, I would like to take 1 to 2 minutes. So during the last week, we still receive a good number of registration. So suppose there are so many participants who discuss the last lecture with the colleagues and friends. So last week we received some more registrations. So it's good that we started with a handful of registration, but now we have more than, I think, 90 students in our Google Classroom right now. So it's good to see. Second is so I posted an assignment. So the main thing about the assignment is not to grading. It's not about the grading. It's just how smartly you understand the concept of the molecular biology, suppose the central dogma of life, or replication, initiation, translation, and so many other things. So don't take it as a grading or some sort of a this sort of number out of 10 or something. So just a way how you understand all the concepts efficiently. Okay. I will upload the key as well, and I just mark or graded your assignments as well. And the third thing, so last time we uploaded the lecture last weekend, so we will upload this lecture as well along with the slides. And at the end of the lecture, Masood will upload the assignment as well. So I'm here right now. So if you have any question, not by asking, but just by tapping, so I will be happy to answer. So Masood, I am giving the floor to you for the rest of the time we have right now. So over to you, Masood. Thank you very much.
Okay. Thank you, Waqaf. So there might be too many slides into this lecture, but I think I might not be able to go through all the slides. So I will send you the edited one later after we finish the lecture. So good evening, everybody. Bangladesh. So here we go. Yeah. So today it's not too many hands-on things here, practical things, but it will be most of them will be theoretical aspects. And how can I get rid of this? Yeah. Okay. So it's on the introduction to bioinformatics and its application, especially in the medical fields and health and public sciences. Yeah. So there is a disclosure I had to declare. And what is bioinformatics? So all of you know, so recently, especially due to the advent of the next generation sequencing, huge amount of data are being generated in the biological sciences. So we need the marriage between the computer science, information technology, and the biological scientists. So that's basically was the impetus for the development of the bioinformatics. So it was defined in many ways by many people and many organizations. And as you know, as I said, because of the production of the huge amount of data, there is an urgent need to make sense of this enormous amount of data and structural data, protein structural data. And as you said, can you hear me properly, or is it?
At least I can hear you properly.
Okay, that's right.
Yeah.
Okay. And other large scale molecular biology efforts. Basically, the bioinformaticians, they use the tools to analyze the data sets and to just give me a second. To reveal the fundamental mechanism underlying the biological problems related to the structure and function of macromolecules, for example, the protein molecules. And for example, you can have biochemical process. For example, there is a genome here, and you have a gene, like a couple of genes here. And in another genome, yeah, in the same organism, there might be lack of some genes. So that ultimately, because these are the two protein products, and there is a deletion of one genes, yeah. So that might ultimately affect the biochemical pathways. That might have an effect on the disease responsible disease process. Sometimes what can happen, you can have the gene duplication. For example, you have one genes and the duplication process is there, and through the when the replication happens, actually, these duplicated genes basically pass through the offsprings, and this can cause the diseases as well. And we also know the SNPs, or the single nucleotide polymorphism, or the mutations in the genes. We are hearing it quite often. Even the like the the even if I go to the like local shops, they will say us, okay, there is any new mutations found on the new variant found in the COVID virus or not. So everybody knows about the bioinformatics now. Okay. So, and the tool of bioinformatics also help us to understand the evolutionary processes. So according to the National Institute of Health, so that's one of the organizations that are involved with the human genome sequencing projects. So they're saying that the research, development, and application of computational tools and approaches for expanding the use of biological, medical, behavioral health data, those acquire you acquire your data, store your data, organize your data, analyze your data, and visualize such data. So doing this all steps actually, so as bioinformatics have been working day and night, has been working tremendously, so to facilitate all the steps. Another definition come from the National Human Genome Research Institute, is that bioinformatics is a branch of biology, yeah. So that is concerned with the acquisition, storage, display, and analysis of information found in nucleic acid and protein sequence data. I agree with this term. Uh, the thing is that actually, so nowadays many biologists are basically trying to become a computational biologist, so you'll be middle in the nowhere actually. So it's better if you learn how to use the tools rather than moving rather than concentrating on how to become a good programmer, how to become a data scientist. Because if you're a good molecular biologist and you know how to use the tools properly, so that's more useful than basically diving into the core computing languages like programming or the software development processes. So now it's possible because we have the tremendous, very good softwares for assemble the genome. So you can look into the individual genomes. You can look into the RNA in the last lecture. Walk us explain very nicely the central dogma. So you can basically analyze the genome along with the transcripts and the proteome, and also the metabolites and the epigenetic changes. We come into later parts of the lecture what the epigenetic changes are. So we can basically analyze, we can do the large scale data analysis using the existing softwares. So the whole process started, the bioinformatics actually, it starts us all with the human genome projects, sequencing projects. So in the 2006, sorry, in the 2000 June, I think it's around this time. Yeah, if you look at this, June 2026, in the June 27, there is a satellite conference between the Bill Clinton and the Tony Blair. They announced the sequencing of the human genome projects, and there are two groups, several groups, I would say, but the two major groups, one is the Wellcome Trust and Sanger Institute in UK, and the National Institute of Health and the Department of Energy in USA, and also one company, Ventnor from the Celera. So they announced the human genome sequencing, completion of the human genome sequencing projects in June 2000. And that involved a lot of money, like, so $3 billion was involved on the 10 years project, but actual sequencing started around, I think it's 1999, and the actual sequencing cost was $300 million for sequencing the one human genome. So that's the information from the Wellcome Trust Sanger Institute, and the seller said there's a $250 million. So, however, you can see the now you can do is in Bangladesh, you can do is $2,000 per human genome. You can do the human genome sequencing, So the cost is reduced tremendously, and I think it could be cheaper if you did like in a commercial scale in from the other commercial vendors. Uh, yeah. So there is a landmark of so the genetics, the father of genetics, Gregor Mendel, started all with that, and I was surprised while I was looking into these data sets actually. So I think I can see it properly. So also the major discoveries were like Frederick Sanger in 1977. He discovered the sequencing method, and Watson and Crick, James Watson Crick, in the 1953. So they determined the structure of the DNA. As you said, the 1977, Sanger sequencing method was developed, and I was surprised to see that the PCR method was developed after the Sanger sequencing method. I didn't know that before looking into this historical overview. And all the sequencing process, the human genome sequencing processes basically evolved between the 1990s and 2003. So there are major events happened. So 1990s, the consortium started working on the human genome sequencing projects, and human genome project in the 1994, the mapping goal achieved, and the first bacterial genome was sequenced Haemophilus influenza in 1995, and in 1997, in 1996, the mouse genetic mapping project started, and the 1997 the joint genome, this is a consortium between uh, NIH and the National Genome Research Institute. They all collaborated for towards the goal to achieve the uh, complete the draft of the human genome sequencing. And also in the 2000 actually, so there is an announcement from the Bill Clinton and the Tony Blair. And 2001, the draft genome sequence was published in the Science and the Nature. And 2002, the mouse genome was draft version of the mouse genome project was completed. Well, I'm mentioning this one because I was lucky to work with the Richard Ems, was one of the uh, what is one of the co-workers in this project. So if you want to look at the better version, so there is a website given, so you can go there and you can download it and you can print it. So recently in Bangladesh, we sequenced almost 100 human genomes. We did the 100 whole human genome sequencing, and everything was done in Bangladesh. And for the quick publication, quick announcement, we published with the four whole genomes, and there's some interesting findings we had. Like we found 11,500 non-synonymous variants. I think what is described what the non-synonymous variants are. So for example, if the DNA changes, variation correspond with the amino acid changes, we call them the non-synonymous changes, and these are deleterious. And we found that some of the genes, the variants are associated with the cardiovascular diseases. And we found the two variants have previously been pathogen factor in familial hypercholesterolemia and the structural disorders. And we also found that some of the markers, pseudoxanthoma elasticum, so that's responsible for the metal dispositions and responsible for the cardiac arrest. I know there are a lot of physicians here, so you know understand better than me these terms. And also we found some genes responsible for cancer progression and some other diseases, including preeclampsia. So growth of DNA sequencing repositories. So you can see the small genome sequencing where it started sequence until the 90s, but after 90 only between 1990 and the 2006 that the only there is only a couple of human genomes sequenced, and after that it increased tremendously. If you go, the vast amount of sequence data has been generated using the, you see there, this much, the vast amount of generated. It's because of the advent of the next generation sequencing processes. You see the first human genome sequencing, it took almost a decade, but now you can sequence using even the high throughput machine, the high-seq or the NovoSeq, you can sequence 48 genomes in including the library prep, you can sequence within 4 or 5 days. And this is a 40 petabytes of DNA were generated in the 2014 and major sequencing centers. And scales of DNA base pair, so one base pair would like 1 BP and 1 kilobase per thousand, and 10 to the power 6 is 1 megabase pair, 10 to the power 9 gigabase pair. These are very important to understand. Yeah. So when you'll be, if you are you'll be working in the human genome data, like thousands of database, sometimes you might basically load up your use up all your repositories. So you might exit. I remember while I was working in the Genome Institute of Singapore, I crossed I almost just the 1 petabyte base pairs. So these are very important to understand these terms. And how much the file sizes can be corresponding to your genome sizes. So this is just for information. Okay. So Central Bioinformatics resources. So NCBI, National Center for Biotechnology Institute, we works with the Ensemble and the European Bioinformatics Institute. UCS is one of the central bioinformatics sites. So all of you are familiar with the PubMed, where you use the PubMed we use for literature searches, entries. So that includes more than 40 databases. BLAST, Wakas will be having a lecture on the basic local alignment search tools. And so there are disease databases, online Mendelian inheritance in men, and the taxonomic databases. You can also look for books and the many additional sources. So we'll be using the NCBI. We'll have some activities right now. So international nucleotide sequence database collaboration. So GenBank, that's maintained by the NCBI. So that's is in the Maryland. That's maintained by the USA. European Nucleotide Archives. So that's maintained by the European Molecular Biology and the European Bioinformatics Institute. So one in the UK, I know that's in the Kingston, Cambridge. So DNA database for Japan. So that's maintained by the Japanese group. The good thing is that, so for example, most popular is the GenBank. Yeah. But if you want a quick submission and if you're struggling with because GenBank will do a lot of queries, yeah. So if you want to submit your genomes and you want to do the quicker one, sometimes I found I might be wrong. It might differ from person to person, but I found it easier to submit through the ENA. And it's all the same actually. So if you submit to the GenBank within the 24, because they collaborate with each other. So if you submit your genome at GenBank, and if you give the date when you want your sequence to be publicly available, so they will, after they're published within the 24 hours, you'll find your genome in all other databases. So you can submit in any of the sequence databases. So this is one of the examples how you can access the NCBI database by the taxonomy browser. So if you go to the taxonomy browser and you just type the Homo sapiens, you can find it's linked with cross-linked with all other databases. For example, the nucleotide databases, express sequence tags, protein databases, structured, and all other databases. So we'll learn as we go along the courses. So we learn all of these things as we go along. Sorry. So taxon represented into a GenBank. So this is a reference from the Pevsner book, Bioinformatics and Functional Genomics. I think it's a 2015 data. So now it might have increased a lot. So you will find the GenBank. So genomes have been deposited from 313,000 species. So have been sequenced from this many of species. Accession number, levels, or sequences. So whenever you submit a sequence in the genome databases, for example, if you submitted the GenBank or the ENA, so for example, when a student gets enrolled in a university or a school, so they get the student ID number. In the same way, whenever anyone submits a genome sequence in the genome databases, they provide us with the accession number. That's the unique identification number for that sequences. So you're using that accession number, you can access your DNA. DNA are the protein molecules. So So GenBank, one of the databases, Genome Quantic DB-SNP is one of the databases, the database of single nucleotide polymorphisms. So you see here, so that's initially they used to follow the 1+5 rules. So that means one alphabet plus 5 digit, but nowadays for the DNA, I think they use the 2+6. So that means if the 2 alphabets followed by the 6 digits. So that's the accession number, and this is the version number. Okay, so whenever you submit a sequence in the databases, they will give you the accession number, and that will be the version number .1. So if you have made any changes and if you updated it, so that will be the version number 2. So that's how the version number gets like changed, but the accession number remains the same. And in the similar way, for so there are two major databases, RefSec and the GenBank. So the difference between the GenBank in the NCBI, in the RefSec and the GenBank is that the GenBank is basically the databases which you cannot change the sequences there, make changes. Any third party cannot make any changes, but the RefSec is the databases. For example, if you have submitted a sequences and someone else worked on the data sets and they found some changes, they want to update it, so they can update the information in the RefSec databases, but not in the GenBank databases. So that's one of the major differences with the GenBank and the RefSec. So biological databases, so we can look for the for example single one gene protein, or we can dig out for the one DNA molecule. For example, you can look for the human beta globulin, there is a gene, and we can also think of the large group. For example, you can look for the all the global genes in the human genome and all the known human variants. You can also look for the variants in the other genomes as well. Or yes, or you might want to study a set of thousands of genes from the different other organisms. So you can think of like you can search for the single gene. You can look for the variation of the same genes in the different other organisms. It will be more clearer when we'll do the blast search. So NCBI gene is a great search starting point. It collects the key information on each gene protein from the major databases, and it covers all the major organisms. And RefSec, as we said, this is curated databases, so third party can basically do annotations or make changes to the GenBank deposited sequences. So NCBI gene, this example of query for beta globulin. So you can go to the NCBI and you can select the option gene. You can type beta globin, and you can find for example, you can find the lots of genes here, beta globin from the different sources. And you can click one of them and you can find the information about the gene. So summary about the genes. So you can go through the stuffs. You can look in the protein databases, and that will give the information about the protein sequences. For example, so this is the whole protein sequence, so that's the size of the amino acid sequence of 147 base pairs, amino acid base pair, and the gene synonyms beta globulin, and you have the cross references with the other databases. If you click on those link, actually that will take you to those other databases. So for example, it can take you to the nucleotide database, it can take you to the taxonomic databases, OMIM databases. So you can, if you want to have a look at the associated information about genes, you can click on those links and you can go. So there are different file formats. One of the most widely used file format we call the FASTA format. So the format is very simple. It starts because you can't like handle the sequence data if you have lots of information along with that. Yeah. So there are some file formats. The FASTA is the most convenient for the bioinformation to use. So it starts the greater than sign, and the first line we have the sequence information, and the second line it starts with the sequence itself. So here this is the amino acid sequences. So you can have the nucleotide sequences as well. So And another feature is the GenBank file format. GenBank file format, along with the header, it has certain other features. For example, if you look into the FASTA is very simple, greater than sign, any of the sequence. But in the GenBank format, you see on the top, I will edit this one. So along with the sequence information, you have the feature actually. For example, yeah, yeah, yeah. For example, this is your genome, and and you have 3 genes here. So let's say it's a forward strand. Yeah. And say, for example, we want to annotate this thing. Yeah. So that's started from 1002 to 2007. Yeah. So this is gene 1, this is gene 2, this is gene 3. Say, for example, my gene 2 is equal to the, so 101007. So that's my gene 2. So we can say that CDS equal to 1000, the coding sequence to 2007, and we can put a gene name here. So that is my gene 2. Yeah. And it's yeah. So this is how you see here the source mean. So this, my genome size is 1 to, say, for example, 10,000 genome size there. So my source is 1 to 10,000. Yeah, you see here. So this is 1 to, this is the size of the genome, and you can put the organism from where it was taken, the source of the organism, molecular type, whether that's a DNA, genomic DNA, or that's a like cDNA from RNA, or yeah, so it's the genomic DNA, and the strain. Your lab provides some identification, strain identification, isolation sources. That's very important, whether that's taken from the human sources or the animal sources, or whatever it is. So as for the secondary symbiont of the, I don't know what this organism is. Sorry about that. And specific host. Yeah. And taxon classification. So every organism has this taxon ID. So I need to provide, and whether within this genome. So because this is a very large one. Yeah, you see here. So almost 4 million base pair. Yeah. So within this 4 million base pairs, so we can express a lot of genes within that. Yeah. And one of the series example is given is actually, that's quite unusual actually. So that should not be like that. So anyway, so 1 to 1395 within these spaces actually. So you have the your coding sequences. So this is how the feature are provided in the GenBank file format. So you can have source information, gene from where your gene starts and ends, coding sequence and start point, stop positions, and it also includes the amino acid translation and also the, okay. So this is the FASTA format. This is a single FASTA format to start with the greater than sign, and you have a nucleotide sequences, and in the single file you can have more than one sequences. Yeah. So you can have one single file in a single sequence in one file, and 3 sequences in the one single file. Okay. There are some activities right now. I think you don't mind to spend 5 to 10 minutes on that one. So if you go to the NCBI, and if you go to the protein option protein, and you can download this one, and you can download in the GenBank format, or you can download the FASTA format, whatever you want. And there is a file format converter. So if you go to this site, you can submit your GenBank file, and that will convert to the FASTA format. So I hope you're, yeah. So is it okay with you, all of you? So there are 2 activities. So if you're stuck, just give me a shout, or you can type in the chat box. If I give you, yeah. Where is the chat box?
Maksud, your screen sharing is stuck right now.
Well, I'm just going for it. Yeah. So,
Okay.
so should I stop this one or? No?
I think you should stop sharing, and if you want to share your browser,
Okay, I'll
then you
just
need to share
okay.
that browser. Yeah.
Okay, I'll just stop share for the time being.
Yeah, yeah, sure.
That's fine.
Maksud, you can continue.
Okay, I'll continue. Okay. So there is another activity you can try at home. So that's open editing frame. And encrypted sequences that contains start codon and stop codon. That's we call them the open editing frame. So that might be coding sequences or may not be transcribed, may not be coding sequences. But so if it's not transcribed, it's just merely we call them the open editing frame. So there is an exercise here. So if you're stuck, just let me know. We'll explain the next class. So you can use these tables to answer your questions. Bioinformatics. So bioinformatics basically integrating the many scientific fields. For example, there is an example here. So what the biologists do, the physicians or the biologists, they hypothesis a question, and microbiologists, biochemists, they do the analysis of their products, and they find out, for example, microbiologists want to use an enzyme for seawater for the production of plastic, and biochemists might do all the biochemists and microbiologists together. Then they can do all the experiments and everything, and they can ask what they want from the mathematician and the statistician, and the computer scientists. They'll formulate the algorithm for the best possible outcome, and the molecular biologists and physicists further interpret and annotate the information, and the bioinformatician integrate all the information into one concept and know all the scientific discipline. So it needs some information in the biological molecules. So we can do the sequence analysis, mutation analysis, gene finding, and RNA sequencing, splice variants. So we look into that, and yeah, protein. We can do the homology modeling, structure, function predictions, and the drug discovery, protein-protein interactions. So there are different units and homology. So this is one of the key biological concepts. So if the sequences are similar, it is assumed that they came from the same ancestor. And say, for example, if you had a look at for example, if the same genes is found in the two organisms, yeah, so we call them the orthologs. And if the gene is similar, homologous genes is found in the same organism, for example, due to gene duplications, that we call it paralogs. For example, CTDP. So it's a cytolethal distending toxin that's produced by the aggregate bacteria and actinomycetocomitans. So if it's found in the same organisms, yeah, we call it orthologs. Sorry, the different organisms, different species, we call them as orthologs. And if it's found the similar genes, CTDA and the CTDB, they're homologs, but they have the different functions, but they're found in the same organisms. We call them the paralogs. So this will be taught in more detail in the BLAST. And in the next lecture, the bioinformatics databases. So you'll be looking at protein databases, disease databases, metabolic databases. There is just some further information. And application, its application in medicine. So developing a drug using the experimental methods takes ages actually. So here, so to rescue the, we can use the computer-aided drug designing molecules, drug designing processes. For example, you can use the homology modeling and structure-based drug designing, and you can do the binding site identification, docking, and through the virtual screening, you can do the compound selection, the lead optimization, and basically ultimately develop the new drugs. So that can help you to personalized medicine. So one of the problems in the cancer, in different types of cancer, somatic mutations. So that means the person is specific mutations. In cancer, it has been found, it has been shown that the mutations are not, you don't find the common mutations in all the patients actually. This is their patient specific mutations, variations are there. So in that case, actually, so personalized medicine, genome combination of phenomics, genomics, transcriptomics can help to identify the discover the markers, and that can help to develop the precision medicine. Predictive medicine, that's another area. So you need to combine the family history, medical, genetics, lifestyle, environment, and combine a combination of all of them nowadays. Bioinformatics work the machine learning. So you can do the deep learning and the machine learning through the machine learning approaches. You can find the best approaches for best preventive measures for the treatment. Gene therapy is another one. So you can do the gene addition, removal of harmful genes, and control of gene expression. Now scientists are working the specifically modified specific areas of a gene using the CRISPR-Cas technology. So I had a presentation in the University of Nottingham on the pathogen genomics. So that's one of the most important areas for the developing countries like, and I've given an example of the country like Bangladesh. So it's a persistent threat to health up to the nation because of the nosocomial infections, pandemics, and the rising spectrum of antimicrobial resistance. So recently I was involved in one of the projects, and I found that all the cholera patients are given the azithromycin, and whenever anyone and we found that whenever anyone's COVID infected, they're also given the azithromycin. So that's a very dangerous thing to do, because that, and so that this indiscriminate use of antibiotic without like proper antimicrobial culture tests, so that they rise the spectrum of antimicrobial resistance. And in Bangladesh, you can go to the many pharmaceuticals and many drugstores, and you can find the antibiotics. I don't know the condition in Pakistan, but so this is a serious issue in our country. We're trying to working on that, and more common infection are the gastrointestinal illness and the respiratory infections. And it basically, the infection varies among the young, old, and the immunocompromised patients, and also be affecting the economy in the developing countries like Bangladesh. And this is the data from the CDC. So the high degree of risk for the developing countries like the bacterial and the protozoal diarrhea, hepatitis A and E, and the typhoid fever, and the vector disease, the dengue fever, malaria, and leptospirosis. And this is today's statistics on the coronavirus cases. So it's almost 17 million, and death is almost 4 million. Yeah. And the second most cause of the death is the lower respiratory infections. And as we said, in the developing countries, one of the problem is the limitations of the existing microbiological practices. Although the microbiology is very useful, but it takes, for example, sometimes it's 48 hours and 72 hours. And for the TB expert from the gene expert, where you don't have the gene expert, and you can understand the time taken to culture the organism and find the antibiotic and to find the appropriate antibiotic for the organisms. And but this can be overcome by the recent advent of the genome sequencing projects. So whole genome sequencing that can give the ultimate resolution between the two related pathogens. So you can find which one is pathogen, which one is non-pathogen. And in the Bangladesh, and in Pakistan, I think there are sequencing facilities available nowadays. And there is one, the nanopore can be applied, nanopore sequencing can be applied in the field level. But the current challenges include the high cost, lack of skilled manpower, and regulatory framework. So in our country, we have to go through the Bangladesh Medical Research Council for approval for every research activities that involves the human subjects. And the technologies are changing, sequencing technologies are changing quite rapidly. So it's very difficult for us to adapt to the new technologies. Internet access limitations is not with broadband limitations across the country, and lack of standard level infrastructure. The brain drain is another issue, and lack of coordination among the research centers is a very big issue in our country, in the developing countries. And low quality of outsourcing. So many labs, what they do, they send their samples, abroad for sequencing and analysis. So during the transportation, so you get the poor quality of samples. So I'll just highlight some of our research activities, what we do on the genomics, because I thought that's really quite useful to know kind of works we do here on the bioinformatics. So this is funded by the Global Challenge Research Fund. So cholera antibiotic resistance in Bangladesh. So what we do actually, so we collect samples from the hospital patients, and from the index patients, we went to their houses to collect the samples from the household family members. And we in the microbiology lab, with the cultural antibiotic sensitivity test and do the DNA sequencing. And combining all this data, we put them in the cloud. And with the help of the team at Nottingham University, we'll be doing the machine learning. We'll be applying the machine learning approaches. So what impact we might have actually? So we can have the increase in the diagnostic effectiveness, sensitivity, reduction in the diagnostic time, and increase in the effectiveness of outbreak. We can do the early detections. So as with the SARS coronaviruses, we hypothesized that there is the Vibrio cholerae, so one of the diarrhea causing organisms that's come into the environment before before it infects a human in a locality like the COVID. So the team, we are working with the University of Maryland, NASA, and the University of Nottingham, using the satellite data and genome data. We hypothesize that at least 90% of the reduction of the time will be achieved to achieve the diagnostic results compared to the conventional cultural methods. And what will happen actually, ultimate impact will be, so we can achieve the sustainable development goal of 1 and 3. That means that they improve the health and welfare of Bangladesh, awareness and adoption of rationalized unified protocols for the data collection, increase the awareness of hygiene and sanitation importance. That covers the SDG 6, and also the sharing of methods and models to fight infectious diseases in other countries. So what bioinformatics helped us? So we recently published a paper on the genotype-phenotype correlation of beta-lactamase of E. coli. So I'll not go into the details. I'll just show you because it's a like sequence type. Yeah. So what it is actually, so we took the leverage, so there is a term called the MLST, multilocus sequence type. So that based on, for example, you have a genome here and have many genes here, yeah? You have another genome, you have many genes. So you take the housekeeping genes, which the same genes from the different organisms. On the basis of that, you do the genotyping. So that's called the sequence typing. So one health issue basically comprises like the organism that circulate that are important for in the environmental perspective as well as the human perspective. So selection of antimicrobial drugs in the healthcare settings that making them the antibiotic resistance. That's the E. coli ST131 sequence step 131 is like the threat to the community, and even in the like the animal health. So due to the use of the indiscriminate use of drugs and antibiotics, they become the resistance and they are becoming a public health threat actually. And they have a clinical impact. They can cause the urinary tract infection and the blood-distant infections. And through the phylogenetic analysis, so ours was the isolate was taken from the urinary tract infections, but we found that the isolates obtained from the urine and the blood samples interleaved. That means there is no distinct cluster. That means so if it's in, that means the isolates taken from the urine, they can also infect. They can also causes septicemia or the bacteremia. So that's where the power of the genomics and the bioinformatics comes. You can isolate through the genome sequencing. You can isolate, for example, like we found some isolates that cause the urinary infection, but that can that cluster with the organism with the isolated from the bloodstream. So immediately, the physicians can be aware using this technology if it's implemented at the field level. So that's okay. So this is a pathogen through the genome sequencing, and we can identify the marker with the marker for potential identification of this kind of. So, and we found that through the genomics, you can quickly identify the markers. Yes, Rajiv Mudundar. Wanted to say something, Rajiv Mudundar. I can't hear it.
Hello, can you hear
Hello,
me?
yeah, I can hear you.
Yeah. Actually, I am also working on the same organism, like E. coli, and that's why my question is that you are comparing those things with the urine infection and blood. But did you check that the some enteropathogenic E. coli, we know that the ETEC, EPEC, they are also responsible for the urine infection or not?
Oh yeah,
Did you check that?
we didn't check
Or
that.
did you check that some avian E. coli are also coming from the poultry industries and introduce some infection in urine, maybe urinary tract infection, maybe gastrointestinal tract infection? Because some E. coli have some E. coli changes its characteristics. Some like we found some new variant of E. coli, which are most commonly found in the poultry, but it is also causes disease in human also. So there is a chance to investigate that the excess route or route transmission route, how to it transmit from poultry to human. So did you check that?
Yeah, Rajiv, thank you for raising this very nice question. Yeah. So we started the lab 3 years back. We started the clinical isolates, and recently we did have sequences from the human diarrheal samples and the poultry samples. We have sequenced them, and yeah, so that's the plan down the line. So what we'll do, we'll put these sequences and along with those sequences to find the similar to do the and so the comparative genomics. Yeah, we can come up. So whether that's at like the potential, but we can find the potential differences among them, whether that's in the gene at the level of gene gain or loss, or it's just, and yes, and we can find whether that cluster with this or, and that will certainly give us an idea actually whether the potentiality of those isolates. at the
I want
genomic.
to add one more thing about that. Did you check the CRISPR regions and prophase region for of those isolates?
Yeah,
Did you found any differences between them?
yeah, Rajiv, I think yeah, our group has checked these things. There are some prophet regions and CRISPR-Cas regions, but it's not provided in this paper.
Okay, okay, okay.
Yeah. Okay, very good. So there is some virulence profile. So we found some of the virulence genes that are present almost all the organisms. And then the question is that if they're present in all the isolates, then what makes the differences in the virulence properties among them? So we looked at the polymorphic sites, and we found that some genes are highly polymorphic compared to the others. And yeah, and we looked at the pathogenic islands. So Rajiv, so this might be answer to your question. So what if you look into the pathogen islands actually, so you find the differences among the isolates. So this is very quite nice study. So we correlated with the bioinformation and the serum. This is the phenotypic data with the genotypic data. And this is a host pathogen interaction data. So this is under development. We're trying to find whether the genotypic differences causes any difference of host pathogen interaction data sets or not. So this was an interesting data set. So for the first time in Bangladesh, we did the sequencing of the Morganella morgani. So initially we thought the three isolates, those are as suspected E. coli, that's cross-reacted with the E. coli 0157. Yeah, then we thought that that's a 157. Then after sequencing, and that was colistin resistant, so we became very excited. We found that colistin resistant 157. But after sequencing, we found that that's a Morganella morgani. And even that's not a bad, because that became a publication, because for the first time we sequenced the Morganella morgani from Bangladesh. So sequencing gives us some nice results, which can be a good publication. And you can see from the bioinformatics analysis, we identified the unique genes and some unique genes in their pathogenicity. Right, I'm not getting the details, so because it will be too early for you. So yeah, so I'll just go through the dengue one. Dengue virus, in the 2019, there is a serious problem with the dengue. You see here. So if you look in the graph, actually, so deaths from 2000 and 2019 reported monthly confirmed and suspected dengue cases. So you see here. So 170 people died due to the dengue reported cases. So you can see the unreported cases how much. So we sequenced some of the genomes from there, and what he found actually. So we found that the increased presence of the potential epitopes could potentially lead to the increased activation from the immune cells, and some of the patients we found the co-infection with the dengue 2 and dengue 4, and re-emerged for dengue 3 in 2017, and rose it drastically for the next 2 years in the 98% 2019. So along with the epitope addition, pre-existing cross-reacting antibodies might also contributing factors. So this is another metagenomics data from, there's something from the chat again. Sorry. So this was quite interesting data. I got a raw data from one of our sequencing centers this is a metagenome data from, what do you call it, SARS-CoV infected people. And you see there's a heterogeneity of the bacterial population. In some cases, we found the large number of bacteria, lactobacillus, and in some cases, we found the abundance of the Moraxella acinetobacter, and see some Staphylococcus. So that means it's not only like SARS coronavirus too. So they can, because they become immunocompromised, they can be infected by any organism, and it depends on the immunity of the person and several other environmental factors, and its physiology and habitats and things. So that means we can find, we didn't look into that actually. So the metagenome, there are some reports, might be, I didn't have a chance to look into that, So there are some reports on whether any correlation with the disease severity or metagenomics pattern or not. So there is a YouTube video. So can you see the video? Last slide.
No video yet. I think you need to change the screen share because it's
Okay.
it's running on the web browser. So I think you need to change that. Yeah, it is shared now.
So just a message for you. Okay, thank you very much. So, yeah. Topic.
Thank you, Maksud, for
Thank
such
you.
a nice piece of presentation. How bioinformatics can help us in discovering new phenomena or by generating or developing new research questions, testing our hypothesis. So, participants, do we have any questions and any interesting comments, suggestions, recommendations regarding any regarding the topic we just discussed? So we are happy to take any questions. So Maksud, are you going to upload the assignment related to this lecture this week, or you don't have any assignment related
I
to
just
this
given a couple of
presentation?
questions. So what I'll do,
Okay.
I'll just I'll just try to give a few more questions so that they can. But you said there are 90 students, isn't it?
Yeah.
I think it's better if we could just give them as an activities.
Yeah,
Like
just
any,
the activity. Yeah, not to
yeah,
grade.
if they have
Yes,
any
just the
problem, they
activity.
can definitely they can
Yeah, I
ask us
think that
right
would be the
away.
best way. Yeah.
And yeah, if you have any issues, you can just write in the blog, as Waka suggested in the last time, so that everyone can participate.
And today's presentation has been uploaded already by Maksud. So so to all the participants, they can go there and they have the presentations already uploaded over there. Okay. If there are no any specific or crisp questions or query, Rajab, yeah, you can say.
Okay, actually, this is my first class. So in previous class, I found some assignment on that. So in two question, I have just I want to just ensure about that. You are talking about the whole human whole genome sequence size. Did you mean that it's a haploid or diploid?
Okay, I can answer this. Actually, I don't want to discuss it right now because in order to because for my next lecture is mine, so I wanted to so I will discuss this assignment in my next lecture for the first few minutes. So I will explain everything. But for your intriguing, I think I just made a quick you can say catch that I just changed that question like this, that 1 byte per nucleotide. So for example, if you have 6 billion bases, so it means
Yeah.
that you have this number of bytes. So once you have this number of bytes, you can easily convert that bytes into the gigabyte or into megabytes or gigabyte. So I just I don't want to go or confuse the participants with the diploid or haploid. Yeah, you are right. So basically, human genome is diploid. So if you download the human genome diploid 2 billion base pair, so it's already diploid. But just to make the things easier, so I just say that okay, you have 1 byte per character or 1 byte per nucleotide. So although it's bits and bytes and so many other things, so I just make it very simple, that just one one nucleotide
I
per byte.
understand.
Yeah.
Okay, thank you.
Thank you. So if there's no other question or query, so we can leave, Maksud, right?
Yeah, we can. Yeah.
Okay. So I need to catch another meeting. So so see you all in our next 3rd Zoom session. So have a nice week.